First thing we do here is the normal pH of body. Normal pH of plasma. Plasma means blood. It is 7.4, which in words we will say, let me write the exact range. It is 7.35 to 7.45. And if we write the same thing in words, it will be called slightly alkaline. So they ask you pH of plasma is slightly acidic, neutral, slightly alkaline. You will say slightly alkaline. If they ask you the exact value, it is 7.35 to 7.45. I hope you remember your chemistry. From chemistry point of view, 7 is considered neutral. So if something is 7.35 to 45, the plasma pH is more than 7, that is towards alkaline. Up to 14, 7 to 14, that is alkaline, and 0 to 7, that is acidic. So pH less than 7 is acidic, pH more than 7 is alkaline, and 7 is considered neutral. So plasma is here somewhat more than 7. That means it is slightly alkaline. Then you have to know another point. pH less than 7.35 is called acidosis, and pH more than 7.45 is called alkalosis. So if due to some disease, due to some abnormality, due to some condition, if pH falls below 7.35, we will say acidosis has happened. If it will go more than 7.45, alkalosis, and the range 7.35 to 45 is normal pH for our body. This is normal pH. Oh, it is written here. Now we will go in little bit into the chemistry. Firstly, what an acid is. You must have studied in your lower grades. An acid is something which releases H positive. So let's say this is Ha is a hypothetical acid. Just an example. Whenever an acid dissociates or breaks, that releases H positive, and instead of writing and, we'll write plus, A negative. So acid is anything that releases a proton. Proton means H positive. H positive ion or proton, that is same thing. Hydrogen ion or proton. So examples would be like HCl. So instead of A, there can be anything. HCl, H2SO4, H2CO3, HNO3. So when these will split, they will release H positive and the other thing will become negative, Cl negative. When this will split, it will give H positive plus NO3 negative, like that. So this is just an example. Anything with any negative ion with H will be present in an acid. Remember, this is the hydrogen ion. And this is the negative ion produced. It is called conjugate base. This is sometimes exam question. They give you any example of an acid and they ask you which of these is conjugate base of this acid. Remember, the negative ion produced from any acid will be called its conjugate base. Chloride is conjugate base of HCl. Nitrate ion is conjugate base of HNO3. So negative ion produced is called the conjugate base. So this is exam question. Normal pH, what is homeostasis? That is exam question. That is first point. Then what is normal pH? That is exam question. Second point. Third point you need to know is what is conjugate base? That is exam question. Then remember, again, this is chemistry, but sometimes they ask you question on this. So just for your understanding, I'm telling. Otherwise, you don't need details. There is something called Ka. Ka is dissociation constant of an acid. Dissociation constant of an acid. It is equal to H positive ion concentration, A negative ion concentration divided by undissociated Ha. Remember, any acid will never fully ionize. Strong acids, they ionize more. Weak acids, they ionize less. So you can see in the reaction, Ha, the acid, it is ionizing into H positive and A negative. So what is dissociation constant? How much this acid is dissociating? That will be concentration of H formed, concentration of A formed, divided by concentration of the undissociated form of acid left behind. That is what is called Ka, dissociation constant. Now, if you take negative log of Ka, that is called pKa. Negative log of anything is called P. From this expression, you can derive the H positive concentration. It's a formula. So if you know all other values, you can find the value of H positive concentration, right? Square bracket means concentration. So negative log of H positive concentration, that is called pH. We keep talking about pH, acidic pH, basic pH, whatever. So P basically means negative log and pH means negative log of H ion concentration. So how much is the H ion concentration? That will tell how strong of an acid it is or how strong of a base or alkali it is. Okay. Because if I right away talk about pKa, you will not know what pKa means. pKa is just the measure of how much an acid is capable of dissociation, how many ions it can produce and how much of it will remain undissociated. Now, the only thing you have to know here is that pH becomes equal to pKa when acid is 50% dissociated. That's it. This is exam question. They ask you, when acid is 50% dissociated, which of these is correct? How can they ask you? This is another exam point you must know. They ask you, when acid is 50% dissociated, which of these will be correct? First, pH is equal to pKa. Option B, pH will be less than pKa. Option C, pH will be more than pKa. Option D, none of the above. They give you such kind of questions. So you know that acid, when it is 50% dissociated, pH is equal to pKa. Or you get question in the opposite form. They ask you, pH is equal to pKa when they give you options A. Acid is 50% dissociated. Acid is less than 50% dissociated. Option C, acid is more than 50% dissociated. Like that, they give you options. So you have to know that when pH and pKa are equal, it is 50% dissociated. And what pKa is, what pH is, that I've told you. Is that clear? Log, if you don't remember, it's just a mathematical expression. Next, we move on to buffers. So this was about what is acid, what is conjugate acid, what is normal pH. These points you must know. Then is buffers. What are buffers? Chemically, buffers are combination of strong acid and weak base or weak acid and strong base. And what their job is, they maintain pH. So in our body, like we did, that the pH of the plasma is 7.35 to 7.45. Let's say we eat something very acidic, the pH should turn acidic. So these buffers, they are various systems in our body. They will not let the pH change. Similarly, let's say we drink something very alkaline, or there are some medicines we have taken which are very alkaline. Still, the buffers will come in, they will play the role, and they will maintain the pH. And what chemically the buffers are, they're combination of strong acid or weak base, or weak acid and strong base. Now there are three types of buffers in our body. These are protein buffers, phosphate buffers, and bicarbonate buffers. Firstly, talking about protein buffers, these are intracellular buffers. That means they function within the cell. So if this buffer goes wrong, if something is wrong, just one cell will be affected, which is not a big deal for a body made up of millions of cells. Even if one cell goes wrong, that will not make much difference. So it is not very important in maintaining the pH as such, but this is one of the buffers. And why proteins act as buffers? Amino acids can be acidic or basic. Remember we did acidic amino acids, glutamic acid and aspartic acid. Basic amino acids were histidine, arginine, lysine. So when these amino acids, they combine to form proteins, this combination can form. Combination of strong acid, weak base, or weak acid, strong base. If this combination comes up, protein can act as a buffer. Now they ask you an important exam point regarding this. They ask you at physiologic pH, which amino acid is maximum responsible for protein buffering capacity? Remember, at physiologic pH, protein, sorry, at physiologic pH, histidine, amino acid histidine is maximum responsible for buffering capacity of proteins. At physiologic pH, it is histidine which contributes maximum. This is the one maximum responsible for buffering capacity of proteins. Then is phosphate buffer. Nothing much important about phosphate buffer. All you have to know is that it is also intracellular, so it will not have impact on the whole body, just one cell. And it is functional in blood cells and kidney tubules. It is present majorly in blood cells and kidney tubules. That's it. Third is bicarbonate buffer. This is most important. It is extracellular, so it will affect the whole body. It is functioning in plasma of blood, and you know blood is circulating all throughout body. So whatever changes this buffer will bring, that will affect the whole body. Why? Because it is functioning in plasma or blood. How this bicarbonate buffer functions, that is very important. Carbon dioxide, you know the carbon dioxide produced in metabolism, the one which we exhale, it combines with water of the blood to form carbonic acid. This is carbonic acid. But you know acids ionize in solution form. So this will further ionize to form H positive, two H positives because it is H2 plus CO3 2 negative. So CO3 2 negative is the conjugate base of carbonic acid. I told you negative ion produced from an acid is called its conjugate base. Now, this reaction is going in both directions. This is how it maintains the pH. pH is what? Negative log of H ion concentration. So, this buffer has to maintain pH means this buffer has to maintain the H ion concentration. How it will do that? Let's say H ion concentration is falling less than normal. So more of carbon dioxide will dissolve in water to form carbonic acid, and more of carbonic acid will produce more of H positive. That means we will exhale less carbon dioxide. Whatever we usually exhale, we will be exhaling less than that. More of the carbon dioxide will be retained in blood itself, and it will form carbonic acid, producing more H positives. So when pH fall in pH will cause the reaction to go in this direction. This direction is when the pH fall. Similarly, if lot of H positive are getting collected here, when pH will rise, here the pH rises. Secondly, when lot of H positives are there, lot of H positive means lot of acid. That means the pH will fall. So when pH fall, which means the H ion concentration has increased, lot of H positives are here. So these H positives will combine with bicarbonates and they will form H2CO3. H2CO3 will form water and carbon dioxide, and we will exhale the carbon dioxide. You know, breathing can be regulated, it can be slowed down, it can be made fast to some extent. So depending upon we need more H positive or we need less H positive, the reaction can go forward and backward. If alkaline pH is coming, if the pH is rising, that means H positive ions are falling. Then carbon dioxide will be retained in body. It will become carbonic acid, which will give lot of H positive. But if pH is falling, that means already lot of H positive is there. Then the H positives will combine with carbonate to form carbonic acid. They will form carbon dioxide and we will exhale the carbon dioxide and throw it out. So brain and the respiratory system will regulate that they have to preserve carbon dioxide or exhale it, or how much to preserve and how much to exhale, and that is how they will regulate the H positives. But they will keep the H positive ion concentration constant. Is that clear? This is what is bicarbonate buffer. Now, let's talk about acidosis. What is acidosis? It is when pH falls below 7.35. That will be called acidosis. What can be the cause? It is of two types. We have respiratory acidosis or metabolic acidosis. Respiratory acidosis will be when problem is there in respiratory system. First, let's talk about respiratory. Problem in respiratory system. What could be the problem? Any respiratory disease. It can be tuberculosis, pneumonia, lung abscess, asthma, anything, any respiratory problem due to which the respiratory system is not exhaling enough of carbon dioxide. It is not getting rid of the carbon dioxide. It is not able to take in enough of oxygen and give out enough of carbon dioxide. Problem in respiratory system which is causing accumulation of carbon dioxide in body. Due to some disease in the respiratory system, carbon dioxide is accumulating in body. When the carbon dioxide accumulate, it will become carbonic acid. It will dissolve in water to form carbonic acid. Carbonic acid will give lot of H positive and CO3 negative. So this increases, the acid will increase. Acid increasing simply means acidosis will increase. H positive ion increases. Lot of H positive is just indicator of lot of acid. So you just have to know that carbon dioxide dissolves to form carbonic acid. Lot of carbon dioxide means lot of carbonic acid. More acid means acidosis. More carbon dioxide means more carbonic acid, and that is what leads to acidosis. More CO2, that causes more of carbonic acid. More carbonic acid means acidosis, right? So if our respiratory system is not helping us breathe properly, it is not exhaling good amount of carbon dioxide will stay in. When it will stay in body, it will become carbonic acid. And more of acid is forming simply means acidosis will happen. This is respiratory acidosis. Metabolic is when problem lies anywhere other than respiratory system. So if anywhere other than respiratory system the problem is lying, that will be called metabolic acidosis. It can be person, for example, it can be some drugs, it can be some toxins. Person has consumed some toxins which are causing acidosis. Person has taken some drugs or tablets which are causing acidosis. It can be increased BMR. BMR is basal metabolic rate. If the metabolic rate of body increases, lot of carbon dioxide is produced. Carbon dioxide is produced in metabolism. So if metabolism becomes fast, lot of carbon dioxide will be there. More carbon dioxide simply means more carbonic acid. So if problem lies anywhere else, any organ, kidneys, drug, toxin, basal metabolic rate, it can be some hormonal problems also which can lead to acidosis. So if it is anything other than respiratory system, that is called metabolic. Is that clear? Okay. And then is alkalosis. Alkalosis is when pH goes higher than 7.45. When pH becomes higher than 7.45, that is alkalosis. Similarly, it can be of two types: respiratory and metabolic. Respiratory, again, if problem lies in respiratory system. For example, hyperventilation. If we are breathing more than normal, hyperventilation, taking lot of oxygen inside and giving out carbon dioxide. So if all the carbon dioxide is thrown on out, very little carbon dioxide will mean very little carbonic acid. And less acid means less acidosis or more of alkalosis. Hyperventilation simply means more oxygen, less carbon dioxide, which will result in less carbonic acid. Less acid means alkaline pH. Hyperventilation usually doesn't happen on its own. It is usually when person is on life support. Let's say person is on some ventilators or some breathing machine, and the machine is switched at a point higher than what the person requires. So usually we can never breathe more and more and fill ourselves with oxygen. Hyperventilation normally doesn't happen. We will just breathe normally, right? Less than normal breathing can happen if there is any respiratory problem, but more than normal breathing will not happen usually. This happens only in hospitalized patients if they are on breathing support, if the machines are excessively inflating them. And metabolic will be when problem lies anywhere other than the respiratory system. For example, vomiting, severe vomiting. You know, stomach has acid. When we vomit, all that acid is thrown out. Again, the stomach will synthesize more acid immediately. Again, the person vomits, the acid is thrown out. Again, the stomach will synthesize acid. Again, the person vomits, it is again thrown out. So constantly acid is being thrown out of body. So body will be left alkaline. So here respiratory system has nothing to do. That is why it is called metabolic. So if the problem is anywhere other than respiratory, it can be same. We have taken some drugs or toxins or poisons which are causing alkalosis or severe vomiting. Anything other than respiratory system will be metabolic alkalosis. Vomiting is a common example. Is that clear? So that's all from your acid, base, and buffers. This is how the pH of the body or the cells is regulated. Next topic we have to do is enzymes. Now we have studied different types of enzymes in whole metabolism process. We'll do the details of enzymes next class, but let's at least do the definition and classification. Enzymes, what is the definition? These are biocatalysts. They increase the speed of reaction. Without the enzyme, the reaction will happen very, very slow. So these enzymes will increase the speed of reaction. Next is classification of enzymes. There are 6 categories. They ask you on this, they ask you which of these is not an enzyme class. There are 6 classes or 6 categories for enzymes. For that, you can remember a mnemonic OATH-LIL that will help you reach to the names. O stands for oxidoreductases. As the name itself describes, these take part in oxidation and reduction reactions. Remember all the reactions involving NAD, NADP, FADH2. I told you that the enzyme name will be dehydrogenase, right? Removal of hydrogen is oxidation process. Addition of oxygen and removal of hydrogen is called oxidation. So all the dehydrogenases, examples are dehydrogenases. For example, pyruvate dehydrogenase. All the dehydrogenases you have studied, they are falling in this category. Then we have also studied oxidase enzyme. These are examples of oxidoreductases. They will deal with oxidation and reduction reactions. And then we've got T. T is for transferases. They will transfer one group from one compound to the other. For example, transferase, the name itself tells they will transfer group. For example, transaminase. Remember we did transaminase, it was transporting amino group from one amino acid to the alpha ketoglutarate. We have just done the protein metabolism. Even kinase is a transferase because it is transferring the phosphate from ATP to whichever compound. For example, glucose becomes glucose 6-phosphate. So the phosphate is being transferred from ATP to the glucose. Remember we did all the phosphate reactions will be, all the ATP reactions will be kinase. So it is a transferase which is transferring the phosphate group. Transaminase was transferring amino group. Then we have H. H stands for hydrolases. They cause breakdown in presence of water. Hydro means water, lases means lysis. Breakdown in presence of water. Example will be digestive enzymes. All the enzymes present in stomach, intestine. For example, amylases, they digest starch and glycogen. Proteases, they digest proteins. Lipases, they will digest lipids. They're all hydrolases. Then L stands for Lyases. Lyases means lysis in absence of water or breakdown in absence of water. Example is aldolase. Remember, aldolase was the enzyme which is causing splitting of fructose 1,6 bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The splitting in glycolysis that is done by aldolase. That was happening without help of water. Then is I stands for isomerases. Of course, it will convert one isomer into other. For example, glucose 6-phosphate getting converted into fructose 6-phosphate. I'm writing the reaction again. So, interconversion of isomers will be done by isomerases. Example of isomerase is epimerase. Remember, glucose and galactose, they are epimers to each other. And in galactose metabolism, we talked about epimerase enzyme, which converts galactose into glucose. And lastly, another L that stands for ligase. Ligases which will form bond or join two things. Join two things or form bond. Can you give any example of ligase you have studied? We have talked about ligase also. Any example you remember? Okay, I give you a hint. It was in the nucleic acids unit, the DNA RNA unit in replication. Anything you remember? What is that enzyme which connects Okazaki fragments together? Remember leading strand, lagging strand. Lagging strand DNA synthesis happen in fragments. They're called Okazaki fragments. And something will come and link those fragments together. No? That was simple. DNA ligase. Something which is linking the DNA, that will be DNA ligase. Okay? There are many other ligases functioning in our body, but the one we studied was DNA ligase. This is the one which was connecting Okazaki fragments of lagging strand together. So these are all different enzyme categories and their examples. So you must know this classification. These are the 6 different types of enzymes. Clear to everyone? So we stop here for now. Next class we'll continue with the rest of the things about enzymes. That's all for now. Study well. See you guys tomorrow. Bye.